Hi, people. We're continuing with cell biology and life science in a nutshell. So, we were learning about um, biosynthesis. So, biosynthesis requires a high level of ATP energy and also consequently low levels of ADP and AMP. Suitable conditions for the simultaneous function of both processes of the processes of um, anabolic processes and catabolic processes, which we learned about in the previous video. So, anabolic being making making molecules, bigger molecules out of smaller molecules, and catabolic processes being the opposite of breaking down molecules. So, um, biosynthetic reactions often take place in compartments within the cell. So biosynthesis is the same as anabolic processes. So you're creating or making molecules um, and within the cells and tissues. So they occur within cell compartments different from those which catabolism occurs. Um, there's a physical separation of energy requiring and energy yielding processes. So, anabolic processes require energy. You use or take up energy from ATP in order to make um, in order to make large complex molecules, while catabolic processes are the opposite. They yield energy. So the energy that comes out of breaking down processes is used for processes of making molecules. So it's like a cycle, basically. Furthermore, biosynthetic reactions are regulated independently of the mechanisms by which catabolism is controlled. Such independent control is made possible by the fact that catabolic and anabolic pathways are not identical. The pacemaker or key enzyme that controls the overall rate of a catabolic route usually does not play any role in the biosynthetic pathway of a compound. Similarly, the pacemaker enzymes of biosynthesis are not involved in catabolism. Catabolic pathways are often regulated by the relative amounts of ATP, ADP, AMP in the cellular compartments in which the pacemaker enzymes are located. So, depending on the state of the cell and the energy levels and the amounts of ATP, ADP, and AMP, you have anabolic or catabolic state occurring within the cell. In general, ATP inhibits and ADP or AMP stimulates such enzymes. So basically, ATP again is used for making molecules. So in anabolism, ADP and AMP are usually at higher levels when you doing catabolic reactions. So these two reactions are in a cycle and they are opposite as far as the ATP levels, which need to be high in anabolism and low in catabolism. And ADP or AMP are the opposite, low in anabolism and high in catabolism. In So ATP would then inhibit catabolism while stimulating anabolism. And this is just what occurs. In contrast, many biosynthetic routes are regulated by concentration of the end products of particular anabolic processes, so that the cell synthesizes only as much of these building blocks as it needs. So, the supply of biosynthetic precursors: when higher animals consume a mixed diet, sufficient quantities of compounds for both biosynthesis and energy supply are available. Carbohydrates yield intermediates of glycolysis and of the phosphogluconate pathway, which in turn yield acetyl coenzyme A or acetyl CoA, um, and also lipids yield glycolytic intermediates and acetyl coenzyme A as well, and many amino acids um, form intermediates of um, both the TCA cycle and glycolysis. So each of these kinds of molecules, carbohydrates, lipids, and amino acids, uh, which come from proteins taken in, um, yield molecules that are used, which are broken down and used to make other complex molecules. Um, Any intermediate withdrawn for biosynthesis can thus be readily replenished by the catabolism of further nutrients. The situation does not always hold. However, microorganisms in particular can derive all of their carbon and energy requirements by utilizing a single carbon source. The sole carbon source may be a substance such as a carbohydrate or a fatty acid, or an intermediate of the TCA cycle, or a substance readily converted to one. In both cases, reactions ancillary to those discussed thus far must occur before the carbon source can be utilized. So this section is metabolism or ATP synthesis, which goes along with that, and this occurs in the mitochondria of cells. In order to understand the mechanism by which the energy released during respiration is conserved as ATP, it is necessary to appreciate the structural features of the mitochondria. These are organelles in animal and plant cells in which oxidative phosphorylation takes place. So oxidative meaning using oxygen. Phosphorylation is adding phosphorus groups. There are many mitochondria in animal tissues, for example, in heart and skeletal muscle, which require large amounts of energy for mechanical work, and in the pancreas as well, where there's biosynthesis, and in the kidney, where the process of excretion takes place. Mitochondria have an outer membrane which allows the passage of most small molecules and ions, and a highly folded inner membrane called crista, which does not allow the passage of even small ions, so maintains a closed space within the cell. The electron transferring molecules of the respiratory chain and the enzymes responsible for ATP synthesis are located in and on this inner membrane. While the space inside, or the matrix, contains the enzymes of the TCA cycle, tricarboxylic acid cycle. So we're going to continue in the next part. Thank you.